it's my pleasure to introduce the second speaker. Andrew Potterton is the head of the platform and Celeris, where he's responsible for all technology efforts within the company. Prior to joining Celeris, Andrew was an benevolent AI, where he completed his PhD, and he completed his PhD at the Institute of Structural and Molecular Biology at the University College London and Evotech in computational chemistry. Thank you very much, Andrew, for joining us, and we are very much looking forward to your seminar.
Thank you for your introduction. I hope everyone can hear me. So I'm excited to give this talk where I'm going to talk about different technology efforts we have at Celeris to drive structural-based design of different proximity-inducing compounds. So just a little bit of what I'm going to talk about. So this talk is split into 4 sections. Just a quick overview of an introduction and just an overview of our ternary complex prediction pipeline method. And then just general uses of such a method, how you can validate such a method, and then a slight aside onto our linker generation tool. Yeah, so this is just a very general overview side, but at Celeris, we develop tech-driven approaches to accelerate drug discovery, and we focus on different types of proximity-inducing compounds to tackle druggable targets in general, but in today's talk, I'll focus on heterobifunctional proteolysis targeting degraders, commonly referred to as PROTACs. And in general, we look at key interactions between the proteins and the compounds in designing and in our design phase. So just an overview of the pipeline. So this pipeline is running essentially 3 stages. And we call this pipeline the PPTCP, so physics-based ternary complex prediction pipeline. So as ternary complex prediction stability has been identified as a major contributor towards degrader-based drug candidates, we believe that this is an important piece in designing compounds for this. So the first stage, conformational ensemble generation. So we believe it's very important to have different conformations of the protein, both the E3 ligase and the protein of interest. So these can be generated using different approaches, so knowledge-based approaches, AI, and more physics-based approaches. So typically, depending on the target, that's what your selection will be dependent on. Obviously, a preference for crystal structures and experimentally determined structures, but failing that, you can use different techniques to generate the conformations you desire, with particular emphasis on conformations surrounding the surface of the binding sites of both the protein of interest and the E3 ligase. Then for each of those conformational pairs, we run protein-protein interaction predictions, so protein-protein docking, in other words. And we take those results and filter them. And then the third stage is running link insertion on those pairs, PPI interactions, and optimizing the complex and scoring, energy scoring, and ranking of the compounds. So just a bit in detail of each of those 3 stages. So first on the conformational ensemble generation, this is more of a standard good computational chemistry, structural bioinformatics approach, I guess, but it's worth reiterating. First is finding structures. Obviously, like I mentioned in the previous slide, you can use different approaches depending on the availability and the types and knowledge and literature you find. Deciding the appropriate domains and whether you think it's worth separating domains depending on their flexibility with respect to each other. Selecting conformations, essentially making the sequence as wild type as possible, and modeling in loops, removing excessive loops if they're modeled in such a way, capping structures for molecular dynamics. That's all on the protein side. On the ligand side, protonation, tautomization, and then bring them to both together. Prediction of both the warhead and the E3 binder binding mode prediction. So this can be traditional approach, knowledge-based approach, or AI approach, depending on the situation. And then protein protonation with respect to the ligand being bound, and then optimization of the ligand binding pose, especially if you're not taking from an experimentally determined structure. That's particularly important. So what I mean by that is molecular dynamics-based refinement. So once you have those conformations, prepared conformations, you're going to run protein-protein interaction prediction. So I've said here in the slide a number of protein of interest conformers and E3 ligase conformers. This can range from, let's say, 5 to 20, depending on the protein, how much conformational flexibility there is, and how much time you have. But essentially, remember, this will be combinatorial, so it's running every protein interest conformer against every E3 ligase conformer. And so exploring the E3 space within those conformers, generating different poses. And then, just to note, when we do that prediction docking, we're including a spacer just to mimic a bit of the link, essentially. Once we predicted all those poses, we filter them based on exit vector distance, depending on the compound linker size, and PPI scoring. So we remove the worst performing scores, essentially, scoring poses, and then clustering, essentially, to not have redundant poses. Once we take all those filtered poses, as shown on the left, we then involve the linker, the PROTAC structure. So as you can see there, this part is all about modeling in the linker. And in some cases, that will not be possible, and that would count as like a failed insertion. And in other conformations, it would be possible. Once, for successful linker insertions, we calculate energies. So there's a bit of preparation, obviously, of optimizing and calculating partial charges, energy minimization, similar to the annealing, and then finally for energy calculation, MMGBSA. And then we use those energies from all those different conformations, so a Boltzmann average of those, to rank different degraders. And we look at all different types of energies, both the energies of the whole complex coming together, ternary complex coming together, but then also the difference between the binary complex and the ternary complex, and on just the proteins coming together, or with and without the PROTAC involved. So that was just a quick summary or overview of our method for ternary complex prediction. So next, I'm just going to talk more generally about uses of a ternary complex prediction pipeline. So this is quite a full slide, but I hope it's interesting. So in the middle is like different stages of drug discovery. And I decided to like think about different use cases of a ternary complex prediction pipeline where, in theory, you could use such a pipeline. And so I'm just going to go for each of them and explain the benefits, how you do so, and some maybe the risks for each of the different sections. So target selection based on feasibility of developing a ternary complex. So in target ID, if you had different targets that you were interested in, you could maybe decide which target is most suitable by essentially doing a virtual screen of enumerated linker libraries with known warheads, if available, E3 binders, different E3 ligases, and then see which one performs the best in the ternary complex prediction pipeline, and then compare the hit rate to other known targets. So either known from your own pipeline or known from the literature as positives and negatives. And obviously this has a cost benefit. If you are successful in achieving this prediction, that you could prevent starting potentially difficult or very difficult or maybe perhaps even impossible projects. And for bigger pharma, rather than picking the right target, it's more about picking the right modality. So is actually proximity-inducing or PROTAC approach suitable for this target, or is something else more suitable? But I would say with this use that there's other factors that could possibly outweigh this, frankly, prediction of the formation of ternary complex, and that things like safety, genetic evidence linked to the disease, opportunity to differentiate, and those might, depending on your company strategy, might have the higher weight. Moving on to a similar but slightly different use is target selection based on selectivity prediction. So if you know for your target of interest or protein of interest that you need to gain selectivity because of opportunity to differentiate over small molecules or safety reasons perhaps, then you can use a ternary complex prediction pipeline to essentially try and predict whether this would be possible. So as we know, selectivity can be achieved using different PPI interfaces. And so essentially what you're looking for is, are there binding site adjacent surface patches that are significantly different between the protein of interest and off targets? And do those differences result in complementary binding with the E3 ligase and not in the off target? So actually, is in this case, we're not considering really the PROTAC, the compound. We're just thinking about the proteins as opportunities for them to come together, and essentially, yeah, looking for opportunity. And then, as the benefit, as I sort of alluded to, is opportunity to differentiate, safety, allowing you to pursue more targets, maybe less severe indications or more diverse indications, and depending on off-target profiling. Next, calculate minimum linker length. So for a given target, you can run essentially a virtual screen of different linker sizes in your ternary complex prediction pipeline, and you can plot essentially number of successful linker insertions against enthalpy. And essentially what we found is that you see a clear drop at a given size in the number of link insertion and in the energy. And then you can use this essentially as a filter, which prevents you wasting time, money synthesizing compounds which are very unlikely to form a ternary complex. Yeah, and as I said, you can use it in filtering both your designs or your linker generation tool. Next, on to 4 and 6. So these are sort of linked together, is basically deciding which compound to synthesize next, but in different stages of drug discovery. So both in hit ID or in hit to lead or lead opt. And the reason I separate these is because I think the differences are going to be larger essentially in hit ID versus hit to lead or lead opt, maybe more subtle changes and changes in different regions, perhaps. And so perhaps the pipeline needs to be run with different settings or different sampling due to that like sensitivity issue, or maybe in later stages. So essentially what you're doing is running on potentially different compounds, seeing which compounds you should synthesize based on the results of a ternary complex prediction pipeline. And obviously this giving you a higher hit rate, cost saving, and especially this is especially useful in later stages because the cost of generating more bespoke compounds perhaps takes more time and more costly therefore. So in that later stage, you probably have increased benefit, but perhaps a harder challenge in terms of prediction of that using a ternary complex prediction pipeline. Number 5 is deciding which compounds synthesize based on selectivity. So this is very related to point 2, but based on the compound as well. So it's seeing if you can design a compound essentially that can drive the complex towards the space which is biased towards your protein of interest rather than the off targets. So you're comparing the energies of that specific interaction, selective interaction versus the off target, and seeing if that is increased with a different design, different warhead, different linker, for example. And that could be like a filtering step in sort of a pipeline. And this could obviously save costs and drive new design, but also save costs of mass spec runs. And selectivity is advantageous for the reasons I said in number 2, so safety and opportunity to differentiate, as well as many other reasons. And finally, which is the most talked about, I guess, is accurate structure prediction. So can you predict an accurate pose, which is essentially experimentally PDB-like or crystal-like pose of your ternary complex? The benefit of this, obviously, is that you can maybe do a rational design once you have that pose and guide 3D SAR. However, I would say that it's likely that, and it's known that proteins form very many different complexes. What is a true form? But that is obviously balanced against the cost of structural and time of structural determination. So we got through the 7 pieces. So now I just want to talk about validation of such a pipeline, and that's with respect to its uses. So in this slide, I'm just talking about the ways you could validate a ternary complex pipeline for different use cases I listed in the previous slide. So I'll go through each of these one by one as well. So for target selection, based on feasibility of developing a ternary complex, you could validate using known degraded targets versus random, essentially not known, not positive targets. And negative targets in general, or true negative targets, may be hard to come by because you might not be able to know if they failed because of ternary complex formation or for some other reason. Of course, there's some mass spec kinase data where they had some positive and negative examples, and so that might be worth exploring for that use case. For target selection based on selectivity predictions, you can use known examples of pairs of proteins where selectivity has been achieved versus random pairs where they have not been achieved. Again, this has the same issue that known negatives are, again, hard to come by. A common problem in this area, I guess. But for calculating minimum linker length, this is quite simple. Essentially, you need a data set with the same warhead, E3 binder, but different linker sizes. Similarly, with which compounds to synthesize, you need compound series data, which is typically what you would get in a project, but there are publicly available data sets of this as well. For selectivity, deciding which compounds to synthesize based on selectivity, you want pairs of data for the same PROTAC, but with different, and the same E3 ligase, but different protein of interest, so 2 different protein of interest at least. And looking at the publicly available data, there's 290 of these pairs for Dmax/DC50 values, and approximately 100 compounds have greater than 5-fold difference in their DC50 values, and approximately 100 again have another 100 have essentially equal values, so they are not selective, and that will give you like 100 positive and negative control. And then finally, for accurate structural prediction, this is sort of obvious. You use the experimentally determined structures, which is an ever-increasing size, but around 15 structures at the moment, right? And this is obviously a very small data set at the moment. And as I said before, about limitations of different conformations revealed by those structures, depending on the different techniques used. Okay, so this is just summarizing how other ternary complex prediction pipelines, at least in the literature, have validated their methods. So I would first like to draw your attention to the histogram. This is just showing the different papers over time for ternary complex prediction pipelines. And you can see it's still quite a niche field, but obviously growing over time. And then the plot on the right shows how different methods validate their methods and how many compounds they validated them against in different classification or correlation or absolute settings. So you can see the majority of pipeline methods predict only against number 7, which is accurate structural prediction. So they only look at whether they can predict the ternary complex structure. And then next is really the 4 to 6, which compounds synthesize. I should point out that triangle is the prospective use case. All the circles are retrospective, so publicly available data and retrospective analysis. And you can see in general, we're limiting ourselves, barring this example of selectivity, around 10 to 12 compounds, especially in these use cases of 3 to 5 and 6. So given that state of play, I want to just talk about what validation we've done internally on our pipeline, which I presented earlier. So I'm just going to present a plethora of different use cases and show what, or potentially illustrate what I think they validate. So this is first a publicly available data set. As you can see the reference on the right-hand side, and for BTK with essentially different linker sizes as seen. And in this, they looked at ternary complex formation in a sort of qualitative way. And we can see with our method, you have two scores, both the energy, as I said before, and of ternary complex formation, and the successful linker insertion as a percentage. And you can see using both of these scores together, you get a nice separation between actives and inactives, and how this trend correlates over the different sets. And essentially what I think this validates is what the shortest linker length is, and possibly a hit ID virtual screen if you were putting different warheads and E3 binders with different linkers, like commercially available linkers in a here virtual screen, you could maybe perhaps decide which of those to synthesize, which of those linker lengths to synthesize, saving you time and money. For a different data set, this is again looking at different linker sizes, but for a small data set for BRD4. But in this case, we're looking at all 4 compounds actually do form a ternary complex, just at different rates. So essentially MZ4, slightly reduced affinity. And again, our pipeline is able to separate that. And again, I think that validates the same 2 use cases. But what's interesting about this is, like I pointed out, it's only a small difference in affinity. But you can see this differences in energies is much reduced compared to the difference between actives and inactives in the BTK set. And then some internal validation, so looking at a virtual screen for a project we're running at the moment. So these are just the positive controls, but you can see in both the successful linker insertion and the energy calculations, we get good separation between the positive and negative controls for the different domains of the protein. And then this is a bit of side, not a validation per se, but just how we're using it in projects. So this shows like how we're using different energy values to go from different medchem designs to running the compounds to selecting for synthesis. So combining all those results together and using relative values, so the maximum, dividing by the maximum essentially for both energy and successful insertions, you can see we can actually compare between different proteins. So the different shapes are different proteins and the different colors are actives and inactives. So you can see we have good separations between actives and inactives between different proteins, but also within each series, you get a nice trend between those 2 different values. And with this overall result, you can see the comparison to the other literature, which I presented on that plot a few slides ago. Okay, onto a slight aside in my final few minutes on linker generation. So this is the more AI-focused part of the talk. So generative chemistry for generating compound designs has been around a while, but specifically for linker designs, there are tools, but I think the people who are familiar with these tools know that the real value of these tools, or the real money essentially, is can you get a scoring function to essentially generate actual decent compounds. And this comes with a lot of use of such a tool, both working with med chemists and comp chemists to understand how they use the tool, but also how med chemists feedback on the tool. So as well as this reinforcement learning within the tool itself, I think there's a sort of similar loop between scientists and different groups of people, which is equally as important. And equally as important as that is the data that goes into that model to train it. So, and here we looked at different sources of data, and this is just some of our pipeline to show how we filtered that data. This is just a quick look at the different sources of data and how that source of data overlap with each other. And that's just the publicly available data, of course. And then just some examples. As I was saying, there's always this balance of creativity and novel versus stable and synthesizable. And this is a push and pull. You have to learn with a model how to generate. And the one way you can do this is looking at commercially synthesizable blocks, synthesizability scores, and pushing the model essentially to develop compounds which are more suitable.